Okay, so what my lab focuses on is the area called the metabolomics, and I will tell you a little bit about metabolomics in a moment. But the idea here is we want to improve biomarker discovery, and we want to use this either in clinical diagnostics as well as toxicology. I come from Concordia University. Lots of people are not familiar with Concordia. It's in Montreal, and it's a relatively young university from 1974, and it's expanding quite rapidly over the last couple of years with lots of research in genomics, synthetic biology, as well as preventative health. So how does the lifestyle influence our health? So coming back to metabolomics, what is it? So it's the comprehensive study of all metabolites present in a given sample, whether this is a biological fluid such as blood, or a cell sample, or a tissue sample. The size of human metabolome is not known, but is estimated to be about 40,000 metabolites, and this is both endogenous and exogenous. And okay, sorry. The next thing you can ask me is why do we want to look at all these metabolites at the same time? And the reason there is these pathways are highly interconnected. So measuring only one of these species really doesn't tell us much about what is being dysregulated in this sample. However, even when we use these state-of-the-art protocols in metabolomics, we run into a big problem, and that is that the low abundance metabolites will not be detected. And these metabolites include everything from hormones, vitamins, so things that are vitally biologically important, so we cannot answer biological questions that we want. This is where my lab comes in. So what we want to do is we want to improve the metabolome coverage and accuracy of this type of analysis. We use many different tools for this, including sequential analysis, new extraction materials, and microextraction. So I'll just give you a very brief flavor of each of these three. So the common method in global metabolomics is to precipitate a plasma sample with methanol and then run it with 4 or 6 different analytical methods. However, this causes a lot of overlap in the analytical results, and the much better approach would be to extract same sample multiple times to avoid this overlap. You can ask me then, well, why don't we just throw different extraction methods onto the same sample? And yes, if you do this, you can achieve some improvement in metabolome coverage. In this particular example, I can see extra 900 metabolites if I quadruple my workflow, so if I use 4 methods rather than 1. This is where new extraction materials really can come and help us. So one of the materials we are looking at is ionic liquids. So here, if I use an ionic liquid, a particular type, in a single extraction step, I can improve my metabolome coverage by 2,000 metabolites, so much better than I did with my previous 4 methods. So there is lots of opportunities for us to improve this. The third type of approach that we are looking in the lab is called in vivo solid phase microextraction. And if you hate going to the doctor to have your blood taken, this is the method for you. We don't take any blood or tissue sample, so no biopsy or blood taken. And the idea is it's a simple hypodermic needle, and the tip of this hypodermic needle is coated and exposed to what you want to sample. You sample it for between 30 seconds to 2 minutes, and then you just take the needle out and analyze what's on the needle. So very non-invasive approach. You don't need 10, 20 milliliters of blood taken or more to run your clinical test. It has many advantages, but one of the ones I want to emphasize here is we can start to detect unstable metabolites, so metabolites that cannot survive once we take the blood out or the tissue out from the sample we want to study. And this is just an example where we are looking at beta-NAT. We can only see it using this in vivo sampling. As soon as we took the blood out, we can no longer detect this metabolite. The tools that we develop, we use them for a variety of applications. One of the studies that we are looking at is the effect of diet on plaque formation in atherosclerosis. Here we are looking at the effect of high protein, low carbohydrate diet and doing time course type of studies to elucidate mechanism. An easier study to explain in 2 minutes is, can we use some of these tools in order to apply them for mycotoxin biomonitoring? So mycotoxins are widely present in food supply worldwide. In Canada, it's estimated that about 75% of food supply contains one or more of these mycotoxins, but there is no good methods to monitor population for these. So my lab set out to develop a method for 20 of these mycotoxins. This is an example of the separation that we can see. We can analyze all of the 20 species that we are interested in. We also did a lot of optimization because we need very high sensitivity. These species are present typically in picogram per mil levels. This is an example of the optimization and sample preparation that we did, and we found good methods that work for 17 out of the 20 mycotoxins that we set out with. For the other 3 that are highly acidic, we need separate methods to achieve the sensitivity that we want. Another issue that we encounter in this mycotoxin work is that mycotoxins can be metabolized by our own bodies once we have ingested them. So any very accurate monitoring should include these metabolites. However, they're often not commercially available, so we run into a problem. How do we look for something that we don't have standards of? So there we are building an in-house library using metabolism incubation studies. So in conclusion, so far we have developed a highly sensitive method for 17 out of 20 metabolites in human plasma. This is the first method of its kind to cover as many mycotoxins as I'm talking about here. And our next step there is to do a population study in Canada to see what our exposure is. And overall, my lab does both targeted and untargeted metabolomics as well as new device development. And the idea is really to improve metabolome coverage and the information that we get and to improve biomarker discovery. So none of this work could be possible without the efforts of my graduate students. So this is them shown back in the summer, and also without the support of the funding agencies. So we are supported by CQDM, NSERC, FRQNT, as well as Brain Canada. Thank you for your attention, and I'm happy to answer questions.
Thanks. So out of your mycotoxins that you tested, did you test aflatoxin?
Yes, we have 4 aflatoxins.
Okay.
Yes.
And is it present? Have you done some real tests?
I know we are right now collecting the exposure study samples. So we are working with the Cartagena Biobank that's going to give us more than 1,000 Canadian samples to test. So that's our next step.
Okay.
Aflatoxins in Canadian samples are not highly prevalent. The most common mycotoxins that we believe we will be finding are deoxynivalenol and ochratoxin A.
Okay. So the next question that I have is like when we follow fermentation, do fermentation to get to a target molecule of interest. Is it possible to study through metabolomics the inhibitors which could be present and which, do not lead to the optimal yield of the target molecule that we have?
Exactly.
Is it rapid through metabolomics to do this study?
The metabolomics can definitely be used. In terms of is it rapid or not, that depends a little bit exactly what pathways you're trying to study, how well the methods developed are for those pathways, things like that. So it's possible, it can be easy or hard, depending on exact application.
Thank you.